So good evening. My name is Milagros Marin. I study biochemistry, but I also did my MSc in translational investigation and personalized medicine. But then I make a jump to a computational neuroscience lab in the University of Granada in Spain. So my research addresses the creation and the study with computationally efficient cerebellar neural models reproducing characteristic spike response patterns. The cerebellum is a key brain structure for the sensory motor control, but also for non-motor functions, including cognitive and emotional processes, such as attention, language, or emotional behavior, and even for non-somatic visceral responses. Cerebellar lesions can contribute to pathological syndromes such as ataxia, schizophrenia, and autism. Granule cells are the smallest but most abundant neurons in the mammalian brain. They regulate information transfer along the major afferent system to the cerebellum. Several of the characteristic firing patterns, such as resonant firing or bursting, are related to the learning processes, synchronization, and rhythmicity in the cerebellum. So, the use of pharmacological drugs together with electrophysiological experiments allows the development of biophysical complex or detailed neural models such as Hodgkin and Hatchley model neuron. But these models requires a large number of differential equations for every ionic conductances to be solved at each time. So, the simulation of these detailed models requires, it's so computationally expensive. What is more, the simulation of millions of these detailed models or complex models is still far from being possible even in the most powerful supercomputers. One alternative to the use of these biophysical detailed models is the reduction in terms of differential equations and parameters to a much simpler neural model with similar dynamics from the detailed model or experimental data. For example, the use of point process neuron models, such as ADEX model or generalized leaky interpretant fire model. However, this fine adjustment of this neuron model parameters is not trivial at all. Optimization algorithms allow the automatic tuning of a set of free parameters given a fitness functions which capture the main features from this detailed model or experimental data that we want to reproduce. So, simplifying this phenomenological neuron model, but identifying and keeping their functional characteristic is a complex process, but is our objective. We addressed a simplification process in which an adaptive exponential interpretant fire model has been adjusted, tuning its parameter, using evolutionary algorithms, but keeping this functional characteristic from the detailed model or experimental data. So in conclusion, the advantages of using these point process neuron models is, firstly, they are much lighter in computational load than realistic models. Secondly, the simulation of large networks of these these simplified point neuron models, such as ADEX neuron, can be addressed even in modern PCs, personal computers. And finally, this increase in efficiency can be more drastic if we do it with much more complex neuron models, such as Golgi cells or Purkinje neurons in the cerebellum. So this automatic creation and tuning of a simplified model or point process neuron model, but keeping this functional characteristic and behavior from the complex model or the detailed model will permit or allow evaluating the behavioral impact of the neurobiological substrate, studying cell properties, network topologies, and also adaptation mechanisms. And finally, the long-term applications applications can be addressed in the framework of neural rehabilitation schemes, reverse engineering, efficient computing, but also in the study of acting principle in biological tasks, studying the neurobiological disorders associated, and thus facilitating treatment for for related mental disorders. So we encourage you to contact us if you are interested on the use or creation of efficient neural networks from these detailed models or your experimental data. So thank you.